Differential functions of heme. We are all familiar with heme as a tightly bound prosthetic group of a number of proteins. Such proteins are called heme protein. The basic structure of heme remains the same in all the heme proteins, similar. There are four pyrrole rings which are joined covalently with iron at the center. So there are four pyrrole rings with iron at the center. This is structure of a pyrrole ring, and this is the shorthand notation of a tetrapyrrole, four pyrrole ring. It is called cyclic tetrapyrrole because they are arranged in a cyclic fashion. And as we said, there is iron in the center. At the pointer shown, there is an iron atom, which could be in the ferrous form or in the ferric form. So there is iron in the center. Now, differential functions of heme. The same heme is performing variety of different roles in various heme proteins. Here I have enlisted some of the heme proteins: hemoglobin, myoglobin, cytochromes, and certain enzymes. Enzymes like catalase, tryptophan, pyrrolase, etc. In hemoglobin, for example, we are talking about now differential functions. In hemoglobin, all of you know what is the function? The function is transport of oxygen. It binds with oxygen. Let me say one thing. None of the common amino, although you know hemoglobin, it has got four polypeptide chain, two alpha and two beta, which are 141, 146 amino acid long. But none of the common amino acids in these chains has got any binding affinity for oxygen. So they have to employ a non-polypeptide component, and that non-polypeptide component is heme. So hemoglobin binds with oxygen. Myoglobin also, it is an oxygen binding protein, but their binding affinity for oxygen, and accordingly their roles are quite different from each other. For example, myoglobin, it has got a very high affinity for oxygen as reflected by such hyperbolic curve. The P50 value, P50 means the partial pressure of oxygen. You see, on x-axis it is partial pressure of oxygen, on y-axis it is percentage saturation. So P50, the partial pressure of oxygen at which it is 50% saturated, that is very, very small, just about 1 torr. Whereas in case of hemoglobin, it is 26 torr. What do these observations indicate? Hemoglobin has got much less affinity for oxygen compared to myoglobin. And the oxygen dissociation curve for oxygen, for hemoglobin, accordingly, it is the sigmoid shape. So much less, 50% saturation at this much. So I have dwelt at length about this oxygen dissociation curve, but sum and substance for what I have said so far, that the same heme, it is binding with oxygen with both in both the proteins, hemoglobin and myoglobin, but the properties are entirely different. So we can also say that heme is performing two different roles in hemoglobin and myoglobin. Now coming to cytochromes. Now cytochromes, they transmit electrons in the electron transport chain B, C, C1, AA3. They are involved in transmittance of electron from one carrier protein to another, from one cytochrome to another. It is the heme which is responsible for transmittance of electron. And during that, the heme iron, the iron which is present at center of the tetrapyrrole ring, it rapidly undergoes ferrous to ferrous, ferric to ferrous interconversions. Ferrous, ferric, as the electrons travel down. But now an important question. Is this possible in hemoglobin? No, it is not. अगर हीमोग्लोबिन को आपने फेरस टू फेरिक कर दिया, it will be converted to a dysfunctional form called methemoglobin, which does not have any oxygen binding properties. So, just an important thing, in cytochrome, ferrous ferric conversion, interconversion is basis of its role as an electron transporter. Whereas in hemoglobin, ferrous को ferric कर दिया, it will be converted to a dysfunctional form. So that again brings out the same point, that the same heme is performing different roles in different heme proteins. Likewise, in cytochrome P450, it is involved in the detoxification of drugs. And then certain enzymes, for example, catalase, tryptophan pyrrolase, इनमें भी हीम होता है. So we repeat, same heme is performing different roles in different proteins, and that all depends upon the apoprotein portion. So we say prosthetic group, it is attached to the apoprotein to give rise to the holoprotein. You are well aware of this equation. So the same prosthetic group performs different roles in different proteins, and the determinant of that is apoprotein. Apoprotein in hemoglobin is different from that of myoglobin, therefore different functions, and same for cytochromes and the enzymes also. Thank you so much.